Okay, today we are diving into some of the most important libraries ever built. But they don't hold books. They hold the blueprints for life itself. We're talking about the massive digital collections known as biological databases. So let's crack open these digital vaults and see how they are absolutely changing science as we know it.
So the whole story of these databases really starts with a problem. And I'm not talking about a small problem. I mean a full-blown data tsunami. In the last few decades, our ability to sequence DNA and analyze proteins, well, it's just exploded. And it's created this almost unimaginable flood of information.
To give you some perspective, just look at this number. By 2012, that was a while ago, just one single DNA database, GenBank, already had over 411 billion base pairs logged. That's billions of A's, T's, C's, and G's, the very letters that write the code of life. I mean, if you tried to print that out, you'd have a stack of paper taller than a skyscraper. And since then, the growth has just been off the charts, truly exponential.
So this incredible amount of data creates a huge challenge, right? When you're literally swimming in an ocean of information, how on earth does a researcher find that one specific thing they're looking for? How do you connect the dots to maybe cure a disease or just understand how an ecosystem works? Doing it by hand? No way. It's impossible. It wasn't always like this. Go back just a couple of decades, and getting this kind of data meant a trip to an actual physical library. You'd have to find the right hardbound journal, probably blow some dust off it, and make photocopies. Today, with the sheer volume of information we have, that method would be like trying to empty the entire ocean with a teacup. It just wouldn't work.
So how did the scientific community tackle this? Well, they started building a completely new kind of library, one that's global, totally digital, and interconnected. Let's take a look at how this incredible resource actually came to be.
It actually started pretty humbly back in 1965 with a book, basically a catalog of protein information. But things went digital pretty fast. By 1982, Europe had started the very first DNA database, and the US and Japan got on board soon after. But the real game changer? That was 1988. This was the moment they all agreed to share their data using a common format. And that single act of collaboration, it transformed these separate little archives into one incredibly powerful, unified global library.
Now, it's important to know that not all of these databases are the same. Think of a regular library, right? You've got original manuscripts tucked away in an archive, and then you have the summarized encyclopedias out in the reference section. Well, biological databases are organized in a really similar way, into two basic types.
So first up, you have the primary databases. This is the raw archive. It's where researchers submit their data straight from their experiments. It's unfiltered, it's first-hand stuff. But then you've got the secondary databases, and these are more like curated encyclopedias. They take that raw data, they analyze it, and then they add these crucial layers of expert review. This is the step that turns all that raw information into actual verified knowledge.
The key difference here is a single word: curation. This is the human touch. It's the process of adding notes, verifying what a gene does, and linking it to other discoveries. Curation is what transforms a long, boring string of letters into a story about what a gene does or how a protein works. It gives you the context that makes the data really, truly useful.
All right, so how does this all work in the real world? Let's follow a researcher on a typical mission. This will show you just how these databases come together to form this incredibly powerful interconnected digital toolkit.
So let's say our researcher's mission is to understand everything about the human insulin protein, from its basic genetic code all the way up to its role in our bodies. So where do they even begin?
Okay, check this out. First, they'll go to a primary database like GenBank to pull the raw DNA sequence. Simple enough. Next, they jump over to a secondary database like UniProt. This gives them the verified protein sequence plus all these expert notes on what it actually does. From there, they can click over to the protein databank to see a 3D model of insulin's physical shape. And finally, they might use a pathway database, something like KEGG, to see how insulin fits into the bigger picture, how it interacts with dozens of other molecules in our cells. See? Each database gives them another critical piece of the puzzle. This interconnected system has absolutely revolutionized biology. It lets researchers build on each other's work piece by piece. But that data tsunami we talked about, it's only getting bigger. And that brings us to the next great frontier in this whole field. I mean, just look at this chart. The amount of data we're storing continues to grow at a mind-boggling rate. It's exponential. And what's interesting is that storing it all is no longer the main problem. The real challenge is actually shifted.
So the new bottleneck, it isn't collecting the data, it's analyzing it. We've built this colossal library, but honestly, we've only read a tiny fraction of the books inside. As one expert puts it, there's a huge gap because this sequence information is not yet explored. The great challenge for scientists today is to dig through it all and find the hidden patterns, to extract the truly useful knowledge buried deep inside.
And that really leaves us with a final massive thought. In these ever-expanding digital archives, what new medicines, what new understanding of evolution, what secrets about our own biology are just sitting there, waiting for the right person to ask the right question? The library is open, and the exploration, well, it's really just begun.